Let's talk about the synapse. Neurons don't actually touch. Information needs to jump from one neuron to the next across the synapse or synaptic cleft. The synapse is the space between them about 20 nanometers wide. Transmission across the synapse is slow because the neurotransmitter has to diffuse from one neuron to the next. There are many synapses. Here we see a bunch of axon endings synapsing with the postsynaptic neuron. An impulse moves from the presynaptic to the postsynaptic neuron. The nerve endings of the presynaptic neuron are filled with synaptic vesicles, which contain neurotransmitters. So the presynaptic neuron synapses with the postsynaptic neuron. Let's look at this more closely. First step at the presynaptic neuron or the axon terminal is that an action potential arrives, or the impulse arrives at the presynaptic neuron. It causes sodium and calcium to diffuse into the axon terminal. Sodium is part of the action potential, and calcium causes the next step to happen at the synapse. The synaptic vesicles join with the membrane and release acetylcholine, their neurotransmitter, by exocytosis. This process requires ATP and mitochondria. Next, the acetylcholine diffuses across the synapse and attaches to sodium receptors on the postsynaptic neuron. This causes them to open, allowing sodium to rush in, creating an action potential or depolarization in the postsynaptic neuron. Now the acetylcholine has done its job, so we want to remove it from the synapse. Cholinesterase, an enzyme, breaks down acetylcholine into acetic acid and choline. This then can be reabsorbed by the presynaptic neuron. So not only the components, but all of the remaining acetylcholine in the synapse would be reabsorbed, then sent down to the cell body where it is packaged once again into synaptic vesicles and reused. Let's go through these once more. Step 1, the nerve impulse arrives at the presynaptic membrane. Then sodium and calcium ions rush into the presynaptic membrane. The calcium causes synaptic vesicles to merge with the membrane and release acetylcholine, the neurotransmitter, into the synaptic cleft by exocytosis. This requires ATP from mitochondria. Acetylcholine diffuses across the synapse and attaches to receptors on the postsynaptic membrane. Sodium channels open. Sodium enters the cell and causes depolarization in the postsynaptic neuron, or in other words, an action potential. Cholinesterase enzyme enters the synapse and breaks down acetylcholine into acetic acid and choline. This stops the postsynaptic neuron from being stimulated. If this didn't happen, and let's say, for example, it's a motor neuron stimulating a muscle, then that would cause a cramp in the muscle. Neurotransmitter components are reabsorbed by the presynaptic neuron. A neuromuscular junction is the synapse of a motor neuron with a muscle fiber. It causes the muscle to contract. Synapses can be excitatory, which means they cause depolarization of the postsynaptic neuron or create an action potential. This means that the sodium channels on the postsynaptic neuron open, causing sodium to rush in, which makes it more likely to fire. Synapses can also be inhibitory, which means they cause hyperpolarization of the postsynaptic neuron. In other words, the axon becomes more negative. This inhibits depolarization. Potassium channels on the postsynaptic neuron open, allowing potassium to diffuse out, making that postsynaptic neuron less likely to fire. Let's look at our example here. We have 4 presynaptic neurons stimulating the postsynaptic cell. We can see that 3 of them are excitatory and 1 is inhibitory. If they all fired at the same time, then one excitatory would negate the effects of the inhibitory, so those two cancel. So we're left with two positive ones, making it more likely that this neuron would fire. Why do we need inhibitory synapses? To prioritize sensory information. For example, what is more important, remembering that you are wearing clothes or listening to the teacher speaking? If all you could think about was that you are wearing clothes because your sensory neurons were sending that information to your brain consistently, then it would make it difficult to focus on anything else. The other reason for inhibitory synapses is to coordinate movement. For example, the biceps and triceps muscles are antagonistic. So if they were both contracting at the same time, there would be no movement in the elbow. We need inhibitory synapses to relax one while the other one is contracting. Painkillers inhibit transmission along sensory neurons. So even though there is pain, your brain doesn't perceive it because the sensory neurons are being blocked. Let's try some questions. A drug is taken that inhibits the activity of cholinesterase. Remember, cholinesterase was the enzyme that breaks down the neurotransmitter in the synapse, which means the neurotransmitter would stay there, causing the postsynaptic neuron to be continuously depolarized. A student drinks alcohol, which opens potassium channels in the postsynaptic neuron. This would cause hyperpolarization of the postsynaptic membrane because remember, potassium is supposed to be on the inside. If it moves out, that means the inside is going to become more negative. A drug, for example, amphetamine, is taken which blocks the reuptake of the neurotransmitter, which is similar to this first one. If the neurotransmitter stays in the synapse because it can't be taken back up into the presynaptic neuron, then the postsynaptic neuron would be continuously depolarized. Let's try a couple more questions. A drug is taken which prevents vesicles from releasing neurotransmitter, which means there are no neurotransmitters in the synapse. So it would have no effect on the postsynaptic neuron. A drug is taken which activates the same receptors as the neurotransmitter, so opens more sodium channels in the postsynaptic neuron. If sodium channels in the postsynaptic neuron open, that means sodium will rush in, causing depolarization of the postsynaptic neuron. Another thing that would happen in the neuron is that it would remove some of the receptors because it would be overstimulated. Summation is when two or more neurons release neurotransmitters at the same time to cause an action potential in the postsynaptic neuron. Let's take a look at the picture. We have 3 presynaptic neurons, and D is the postsynaptic neuron. And then we have a graph showing the measurement of charge in neuron D. We can see that A and B alone do not reach threshold, but they are causing some sodium to go in, so they are both excitatory. C is causing hyperpolarization, so it is an inhibitory neuron. Notice that when A and B both release their neurotransmitter at the same time, they reach threshold and cause depolarization in the postsynaptic neuron. The parasympathetic and sympathetic nervous system both use 2 neurons and 1 ganglion for each impulse. They both start in the spinal cord, and you can see they go through a ganglion to the ganglionic nerve before stimulating the effector organ. The other thing to note here is that the parasympathetic nervous system uses acetylcholine as its neurotransmitter, and the sympathetic nervous system uses noradrenaline or norepinephrine as its neurotransmitter. So to summarize, the sympathetic and parasympathetic nervous systems, they are both under involuntary control. You don't have to think about them for them to work. Function of sympathetic is fight or flight. Now we say fight or flight, but anything that requires more focus would stimulate more of the sympathetic nervous system versus the parasympathetic. Things like writing a test, walking, running, playing a sport, being more active is sympathetic. Parasympathetic is rest and digest. So while you are resting, there's more blood sent to your digestive organs so that you can digest your food. Finally, the sympathetic nervous system uses norepinephrine as its neurotransmitter, and the parasympathetic nervous system uses acetylcholine